All right, what's up, you guys? So this was actually a paper that I presented in graduate school a couple years ago. It came out in 2021, and I graduated in 2024. I got my PhD technically in May, but I defended my dissertation in March, which is basically the unofficial, graduation. So, but the ceremony was in May. So I got my PhD a couple months ago. So this is a really fun paper. And I'm no longer studying addiction really. I mean, maybe in the future I'll go back to it, but right now I study fluid homeostasis. So I thought it'd be fun to kind of revisit what I was doing in grad school. But basically, the neurobiological underpinnings of drug addiction is shown in this picture right here. So there's a lot of effects biologically and socially and psychologically that produce people that have drug addiction issues. But in the canonical literature of what we think is producing the rewarding effects of drugs, and then two, the outcome of somebody being a drug addict or having substance abuse disorder or whatever, comes from this mesolimbic and mesocortical pathway. So in the ventral tegmental area, which is this brain structure that's pretty far back in your brain, it produces dopamine. Now, when you stimulate this, or in general, you have dopaminergic release, and dopaminergic just means, a dopamine affected system. But when you have dopamine release from the VTA, it goes to the nucleus accumbens, which is a structure in the limbic system that is canonically associated with all sorts of things, but primarily in this context is going to be reward, as well as release to the cortex. Now, the prefrontal cortex is a really interesting area because it's really important for executive control over actions, but it also contacts back to the nucleus accumbens itself. So you have a lot of activity that's happening between these three structures. You have dopamine release to the nucleus accumbens, and it affects these dopaminergic medium spiny neurons in the nucleus accumbens. And then you also have activity in the cortex and excitation of these glutamatergic neurons that then also send projections back to the nucleus accumbens. So it's kind of complicated, but there's a huge interweb of activity between the VTA, the nucleus accumbens, and the prefrontal cortex. And that's why, we call this the reward pathway, because it's extremely important. Now, I do want to point out, because I was talking to a friend of mine who's not in the field, and they were talking about dopamine, and you hear a lot about dopamine being reward, and Andrew Huberman talks about dopamine being necessary for motivation and effort. Excuse me, but dopamine is associated and necessary for all sorts of things. Like if you have too much dopamine or not enough dopamine, then you'll lose control over your motor, your ability to just move around. I mean, that's when people have Parkinson's disease. It's a death of dopaminergic cell bodies in the substantia nigra. So I don't want you just to think that dopamine is only important for reward. It is very important for reward and motivation and all these kinds of things. But also dopamine is just necessary for your ability to fucking move around and pick up a glass of water or whatever. So just keep in mind that when you're hearing people that are talking about these kind of, these really complicated neurochemistry, it's never as simple as it sounds. You know what I mean? It's you have people that are influencers that are popular that talk about dopamine detox and all this kind of thing. I'm not really sure what they mean by that exactly, because we're not getting into that. But the point is that it's complicated, but all drugs of abuse, okay? Alcohol, all stimulants for sure, nicotine, opiates, all drugs that people commonly abuse increase dopaminergic release from the VTA to these other structures. Now, they all do it in slightly different ways. Okay, so you will see throughout this presentation that I am extremely artistic, and you will see my beautiful renditions of various brain regions reimagined as squares and circles. So I'm super bad at making these diagrams, but I think that they get the point across in a logical way. So I'm going to do my best, but you can see that the things that look good, like this brain and this neuron, I stole from somewhere. I don't know. that illegal? I don't know. I got them from some kind of neuroscience resource PowerPoint thing. It was all made in PowerPoint, but I didn't make them. But in any case, okay, so we know that all drugs of abuse, again, nicotine, alcohol, opiates, stimulants, they all in some way, either directly or indirectly, increase the release of dopamine from the VTA to the nucleus accumbens and to the prefrontal cortex, okay, the mesolimbocortical reward pathway. Okay, so how does this happen in the context of opiates? Well, this study in particular is going to be using morphine and not heroin or fentanyl, and that's probably fentanyl. I don't know if that's technically Schedule 2 or something, because I think that fentanyl is actually used in hospitals and in like a medical setting, and heroin used to, can also be used technically, I think, for that stuff, but heroin is Schedule 1. Morphine is a little bit easier to get your hands on, I believe, in a research setting, you know what I mean? So like when I was in grad school, we used oxycodone, and we got that directly from the NIDA drug supply, and it wasn't like a Schedule 1 substance because it's used obviously for pain management. Heroin, I'm fairly certain, is Schedule 1. Morphine, I'm assuming that the reason why they used morphine is because that's just like the easiest thing for them to get a hold of in whatever research context this was. But morphine is working the same way that heroin is working. Okay, so in the brain, heroin just becomes morphine. It's just that heroin is a little bit different because it's a little bit more lipophilic than morphine, so it passes through the blood-brain barrier a lot faster than morphine does, which is why it has a different subjective effect. But when we're looking at like just the overall mechanism, which is what this paper is going to be looking at, it's not too far of a stretch to be saying that although these researchers are using morphine specifically, that we might be able to generalize a lot of this information to other opiates like fentanyl or heroin or oxycodone or whatever the fuck other opiate is that we're talking about, right? So in any case, the way that opiates increase dopamine in this reward pathway is by working on primarily mu receptors. Mu opioid receptors. You may have heard of these things. So these are G protein coupled or metabotropic receptors. That all means the same thing. G protein, metabotropic. It's all referring to the same type of structure, which is this 7 transmembrane protein. Okay, so these little membrane things here are actually sitting on the the cell, and they have part of them is in the intracellular space. If we have like, this is like, dude, how the fuck, if you have never seen this, it's not going to help you for me putting my fucking hand like this, because in my mind, I'm imagining some picture from some undergrad textbook that I read. But in any case, essentially the protein is sitting, like if this is the cell's like fucking lipid bilayer, or like the extracellular fucking, the thing that's separating the inside of the cell to the extra side of the outside of the cell, right? The skin of the cell, whatever the fuck. The skeleton of the cell, however you want to say it. The protein, if this is a protein, it's sitting like this on the cell, and part of it, as we can see here, is on the outside and part of it's on the inside, and the part that's on the inside is coupled to a G protein, and it has all sorts of other things that it can be associated with and then cause downstream effects on the inside of the cell. So that's what a G protein coupled receptor is. That's what a metabotropic receptor is. It's doing all sorts of shit. But morphine is going to bind to these mu receptors, okay? And then the mu receptor is going to inhibit this neuron here, and this neuron is very important because it's a GABA interneuron. So there's a lot, like these papers are so informative, and if you are a undergrad or if you're a graduate student and you're fucking sitting out on journal clubs, bro, you got to go to journal clubs because you will learn so much just about whatever field it is that you're in, because it requires so much background information to really understand these papers, right? So just in trying to explain the paper, you see I get stuck on this just to explain, the basics of what's happening here. So really don't sleep on this kind of stuff. But GABA, okay, so I think I don't remember if I was going to define GABA or interneuron first, but GABA is the main inhibitory neurotransmitter in your brain. Okay, so when you get an increase in GABA, you get an increase in inhibition of whatever target neuron it's affecting. Okay, so we have neurons that are localized in certain brain structures. So in the VTA, for instance, there are some neurons that communicate only to other neurons that are in the VTA. Okay, and this is true of the nucleus accumbens and the prefrontal cortex. And essentially what these neurons are called are interneurons. So you have some things that are called principal neurons, which are projecting to other areas of the brain, meaning that they originate, let's say in the VTA, but then they send collaterals to the cortex, or collaterals, like in collateral just means like a fucking growth off the neuron, right? To the nucleus accumbens. So that's a principal neuron, a neuron that, the soma is in one nuclei, and then it projects to another nuclei, and the nuclei is just referring to a cluster of cells in the brain. Okay, so a cluster of cells in the central nervous system is called a nuclei or a nucleus, and the same thing when it's outside of the nervous system, outside of the central nervous system, but is still nerve cells, is called ganglia. Okay, so this basic vernacular is also really important to kind of get a grasp on when you're like listening to people talk about this, because it will really demystify a lot. So in any case, nuclei is a bundle of neurons within the central nervous system, and what we have here is interneurons and principal neurons. Interneurons are only communicating within that small structure. I mean, it doesn't have to be a small structure, it could be a big structure, but the point is that they don't project to other nuclei, whereas principal neurons do. Okay, so why is this important? It's because these interneurons are inhibitory. Okay, so if we just see this whole thing right here, I wish that I had a green screen that I could fucking point to, but essentially you have this neuron here, the small neuron, and it is an interneuron, meaning that it's only communicating within the VTA, the ventral tegmental area, this dopamine nucleus. And it is an inhibitory neuron, meaning that it communicates to other principal neurons that are in the VTA, and it says, hey, stop fucking shooting dopamine all over the place. All right, we got enough motivation. We're fucking Goggins the fuck out. Like, we don't need any more dopamine. Okay, so this inhibitory neuron releases GABA onto these dopamine neurons and reduces activity. So what happens here is that morphine binds to these mu receptors and inhibits the GABA neurons. Okay, so what does that mean? We have two negatives, so we get a positive, right? So let me say that in a different way, because this gets fucking kind of confusing, is that a lot of times the way that these systems work is through inhibiting inhibitors. Okay, so we have morphine, and it is inhibitory to these GABA interneurons. And the net result of that is an increased release of dopamine to the nucleus accumbens and also the cortex. Okay, so that's how morphine and opiates in general are increasing dopaminergic release to this pathway, and that's the hypothesized mechanism for how these drugs are reinforcing and addictive. Okay, so that's like the basic neurobiology of drugs of abuse and how that system works. That was a lot of information, so I apologize. Probably you can ignore most of it. Okay, so there are behavioral phenotypes that define addiction. Okay, so if you're a psychologist and you're looking at the DSM, or you're a clinician and you're looking at the DSM, obviously there's going to be a lot of things that characterize drug addiction, drug addiction and calling people drug addicts is kind of not the way that things are talked about anymore. But if you're talking about a substance use disorder, I'm going to refer to it as addiction because, you know what, I come from a whole family of addicts. I'm fucking sober, clean addict, so I don't really give a fuck. I'm going to call it addiction. But in any case, addiction is characterized by pharmacological addiction. I don't know anything about behavioral addictions like fucking gambling or gaming or whatever. But as far as a pharmacological addiction goes, it's characterized by two phenomena. One called tolerance. Okay, so tolerance, I think that people are familiar with this idea, this concept, but basically it just means that the more that you do the drug, the more of the drug you will need in order to get a particular response. Okay, so this is also called a rightward shift in the dose response curve, more technically speaking. So this means that as you've done the drug more, it takes more of the drug in order to get the same response. Okay, so this is tolerance. Now, something that's commonly associated with tolerance is withdrawal, meaning that once you try to stop taking the drug, then a lot of negative aspects are going to occur. And that's because your body is always trying to get to a homeostatic set point that it kind of wants to be at. Okay, so it's got some, biological rhythms that are being controlled by your brain. And when you start putting exogenous chemicals into your body, your brain is trying to combat the effects of the drug. So, a really common example of this for opiates would be constipation. And it's really interesting how that works because you actually have opioid receptors in the gut and in the intestines. I don't know much about the gut and the intestines, okay? So let me say that somewhere that is necessary for you to take a shit, you have opioid receptors that halt that gut motility or that intestine motility, okay? So somebody who fucking knows what they're talking about when it comes to the physiology outside of the brain can go ahead and let me know how I'm getting that wrong. But essentially, it halts gut motility and reduces your ability to take a dump, okay? So when people are withdrawing from opioids, they have fucking massive diarrhea because now the thing that was inhibiting their ability to take a shit is gone. And so now their body's like, okay, we gotta fucking take a shit now. Okay, so essentially this effect of withdrawal can be seen in many things. It can also be seen in hyperalgesia, okay, which it means that the patient or the person who is addicted to the drug and is trying to withdraw are hypersensitive to all sorts of various stimuli that we wouldn't normally perceive as painful. But this is an analgesic drug. So, generally speaking, when you're in withdrawal, you're going to get the inverse of whatever the drug was doing to you, right? So if it's something that makes you not feel pain, you're probably going to be in a lot of pain. If it's something that keeps you from having diarrhea, you're going to start to have a lot of diarrhea. Keeps you from pooping, you're going to get diarrhea, right? So this is withdrawal. And the physical symptoms of withdrawal are not the primary reason why somebody is a drug addict, but it really makes the process of getting sober and staying abstinent difficult. Now, there's a whole other side of this once people do get sober and clean that is, outside of the physiological symptoms. And there's also other drugs that a lot of people don't normally associate with physiological withdrawal, and so they think that those aren't addictive, like weed. Weed is absolutely addictive, for sure. A lot of people fucking kill themselves with an addiction to something as innocuous as weed. It's not the weed's fault. It's just that, people that are drug addicts that have a mental disorder, they have a disease, they do things that are, they look, I'm not trying to fucking argue with you about whether or not weed is addictive. Just take my word for it as somebody who sold their car for an ounce of weed and fucking smoked it all, okay? I had this idea that I wasn't, the point is that there's a lot going on here. This is one of them. It's one minute aspect of the whole thing that is drug addiction, right? So we have this tolerance, leads to withdrawal, and then the person tries to get sober and they're abstinent for some period of time, but it's super fucking uncomfortable and they relapse. Okay, so there's a lot going on to addiction also, like sensitization that I'm not talking about. But this paper is focusing on this aspect of addiction. Now, the hypothesis as to why there's negative effects associated with this chronic opioid use, and in this case morphine, has to do with dopaminergic dysregulation, okay? So the idea is that there's chronic morphine, this causes dopamine dysregulation, and this dysregulation of dopamine causes negative symptoms. Now, not the symptoms that I was talking about with the increased pain and the diarrhea, but there's a lot of negative emotional aspects as well to withdrawal, other than just the physical ones. It's all physical. But there's a lot of emotional aspects to it, right? So how does this happen? So this paper, I'm going to show you the whole paper right now in my artistic rendition of squares and circles and lines. So what this paper is going to try to prove to us, okay? So we've gone through a lot already. Like, Jesus Christ, we've gone through so much. We've gone through how drugs are addictive, how they're reinforcing the neurobiology of that. We've gone through some of the behavioral phenotypes of addiction, which is tolerance, withdrawal, the cycle of abstinence and relapse. And this paper is trying to explain one of the mechanisms that contributes to this, which is dopamine is dysregulated somehow, and this causes negative symptoms, and this paper is going to try and show us one of the mechanisms by which this happens. Now, in the central nucleus of the amygdala, that's what the CEA stands for as far as I remember. Again, it's been a while since I've done this paper, but CEA generally means central nucleus of the amygdala. There are neurons that produce a stress hormone, corticotropin releasing hormone. Okay, so this is a stressful stimulus, or it's a stressful neuron, and the hormone that is released causes stress. And there's all sorts of papers that, you know, tie this hormone to this stress response. Now, the idea behind this paper and what they're going to try to show us is that these neurons are recruited through chronic morphine use. They then project to the ventral tegmental area and reduce activity in dopamine neurons. Now, this little squiggle here is just when you're doing electrophysiology and you're looking at the electrical potential of cells, this is kind of what those traces look like. So that's why I put this in here. But I was trying to say a decrease in action potentials, which then results in a decrease in dopamine release, ultimately to the nucleus accumbens. And this dysregulation of dopamine to the nucleus accumbens is essentially very aversive and uncomfortable. So this is what the main hypothesis of the paper is trying to say. Now, specifically, these CRH neurons are projecting to what we'll call a morphine ensemble. Now, the topic of ensembles is, kind of what I studied in my, for my dissertation, and it's very complex, it's very convoluted, and let's just say, it's not that complicated, it's just that there's a lot of nuance and philosophical fucking shit going on here. But let me just say that ensembles are referring to the groups of neurons that are specific to a particular, let's say, learned event, okay? Now, in this paper, what they're referring to are neurons that are sensitive to the physiological effects of morphine. Okay, so ensemble generally has a learning component associated with it, like in the literature, but in this paper, when they say ensemble, they're just referring to the neurons that are activated when the organism is getting morphine. Okay, so we have some group of neurons that are sensitive to morphine, and that is the morphine ensemble in the VTA. Okay, so this diagram right here is the fucking paper. Okay, so we're going to come back to this at the end if I remember correctly. I think I've showed this again. But basically, there's going to be many experiments that are going to be trying to prove to us that indeed the central nucleus of the amygdala projects to the VTA. Specifically, CRH neurons are projecting to morphine-sensitive cells in the VTA, reducing their activity, reducing dopamine release to the nucleus accumbens, and this is what's causing a lot of the uncomfortable things that are occurring with opioid withdrawal. Okay, so this is going to be a little primer for each figure, and each figure is going to be several experiments that are trying to show something. So here I've summarized what I'm about to talk about, which is the activation of ventral tegmental area neuronal ensembles recruited by morphine induces dopamine release in the nucleus accumbens and positive reinforcement. So I think I have some grammatical issues here. Poor Wes. Anyway, okay, so in contemporary behavioral neuroscience, when they're trying to affect some kind of behavioral output in the brain, it is extremely common to be using viruses. And in order to understand, what's happening here, it's imperative that you know how to read a viral construct, a viral, essentially I can't think of the fucking word right now, but essentially their little blueprints of what the virus is, okay? So each one of these things, if you're looking at any contemporary high-impact neuroscience paper, there's a very high likelihood that there's going to be either some kind of viral construct here or a transgenic construct that has some kind of tool that is producing, whatever effect that we're doing here. So let's go ahead and break this down because this is fucking imperative. So we have an ESAIR CRE ERT2 diOM cherry. So what does that mean? ESAIR is the promoter for this virus. So when you're looking to see what neurons are going to be affected by the virus, now, you want to see what the promoter is, because generally the reason why these viruses are being injected, and these are synthetic viruses, by the way, which are generally not harmful. Okay, so basically, I don't know if actually I don't see what this is encapsulated in. If this is this is probably an adeno-associated virus, but essentially they take some kind of virus, they remove the harmful part of it, and they keep the part that replicates the DNA that they want to replicate. In this case, it's going to be the rest of this viral construct here. But in any case, we want to know, whenever you're looking at these kind of viral constructs, you want to know what neurons are being affected. So here we have ECER. Now, ECER, I can't remember what it stands for, but essentially what it's selecting for are neurons that are highly activated. So it's a combination of several different immediate early genes, as far as I remember, that then are activated when the neuron is firing. Not temporarily while the neuron is firing, but the neuron fires and then there's a time course and then you get expression of these proteins. And this promoter is saying, okay, when we see those proteins, we're going to express the rest of what's in this virus. Okay, so there's all sorts of different types of promoters that will select for different things. There's promoters that select for neurons. There's promoters that select for glia. There's neurons that promote that select for excitatory neurons or inhibitory neurons. So yeah, that's very important to understand. Now, CRHCeA is a molecule that results in recombination in genes that are flanked by LOXP sites. So what does that mean? It's complicated, but basically what it means is that this is some kind of enzyme, that will find the rest of whatever tool is being used in this viral construct, and it'll cause that to be expressed, okay? Now, specifically, we have on here a ERT2 that's associated with this CRHCeA. Now, this is super important because it gives you temporal control over when you get the expression of the tool. And whenever I say tool, I'm just referring to the thing that is going to be doing your experimental manipulation for you to see like whatever thing it is that you're fucking with, right? So ERT2 is a synthetic estrogen receptor that's sensitive to exogenous selective estrogen receptor modulators that traffics CRHCeA to the nucleus. So that's a lot of jargon. A selective estrogen receptor modulator is a drug. In this case, it's 4-hydroxytamoxifen is probably what they're going to use in this paper. I can't remember. Again, I haven't read the paper in a long time. But essentially what's happening here is this CRHCeA ERT2 is really advanced and important and gives us a lot of control over how we're manipulating the brain. Okay, so it's fucking phenomenally impressive that this exists. So this is going to make a little bit more sense after I explain what DIO means. So this is a double floxed inverse open reading frame. And that's what these LOXP sites are talking about. It's some genetic shit that I'm not a geneticist, so I only know enough to understand barely how the fucking tool works, right? But basically, when you see in a paper DIO, that means that whatever comes after DIO is dependent on CRHCeA, okay? So in order to get the thing that we care about, which after all of this is just to get M-cherry, which is a red fluorescing protein. Okay, so what that means is that when we shine a light onto this protein, it's going to look red under a microscope. So that's really important because that's how we see the neurons that we're trying to manipulate. So what this whole thing is saying, okay, is that in neurons that are activated, I'm going to express CRHCeA, and when I express CRHCeA, if you inject tamoxifen, I will then activate a red fluorescing protein. Okay, so that's what that says. Okay, in activated neurons, that's ECER, we get CRHCeA recombinase, which is an enzyme. Okay, but you're only going to be able to get that enzyme into the nucleus if you inject tamoxifen because of this estrogen receptor. When you inject tamoxifen, it's going to traffic the CRHCeA into the nucleus where it can then fuck with these LOXP sites and express this tool. So the important thing here is that what this gives the researchers the ability to do is to target neurons that are sensitive to morphine at a very specific time point. Okay, so everyone's probably asleep. I have no idea why anybody who's not interested in this would even be fucking watching this, but regardless, this is what's happening. So we have a ESAIR, CRHCeA plus a Cre-dependent M-cherry. And here's the experimental paradigm here, okay? So, yeah, as I suspected, it is an AAV. And they inject AAVs into the ventral tegmental area, which is shown here in red. And these are the little injectors that they're trying to show us is how they injected the virus. 3 weeks before the experiment starts. Now, they do 3 weeks presumably to allow for proper viral expression. By the way, we're getting into the paper now. All that other shit that I was just talking about was just to explain the paper. And normally, I have no idea how long I've been recording, but normally in a context where this is like a journal club for grad students or for scientists, I would have breezed through all that in about 5 minutes. But I think that for people who are not familiar with this, all that introduction stuff is really fucking important. Just because you probably haven't been, you probably haven't learned about it yet unless you've taken specifically a course for this, right? So 3 weeks before the experiment starts, they inject the AAV, which is an adeno-associated virus that has all of this stuff in it. They then give a tamoxifen infusion on day 1. So it's tamoxifen. It's not 4-hydroxytamoxifen. So that's important because essentially it takes 24 hours for, do I have some kind of shit on my screen? Anyway, they give tamoxifen 24 hours before they want to do the labeling because that's how long it takes for tamoxifen to break down into 4-hydroxytamoxifen. It's not important. Essentially what this is saying is that when they give tamoxifen on day 1, that means that they can give an intraperitoneal injection of either saline or morphine, and then they can tag those neurons that were activated either by the saline injection or the morphine injection with a red fluorescing protein. So then they're going to wait a couple of days, and then they're going to kill the mouse, and they're going to do immunohistochemistry for tyrosine hydroxylase, which is a marker for dopamine. So it's not actually staining directly for the chemical dopamine. It's staining for tyrosine hydroxylase, which is a necessary component for dopamine. Okay, so here is the actual data from this experiment. We can see on the y-axis that we have the number of cells that they quantified. And on the x-axis, they have the anterior-posterior distance from bregma in millimeters. So bregma is an area on the skull where the skull plates meet. So when you're injecting viruses into rats or mice or any experimental animal, essentially you need to know which nucleus it is that you're going to be putting the virus into. And so that's where these coordinates come into play. So what this is saying is that, there is a gradient of these neurons that are sensitive to either saline or morphine. So the saline neurons are a little bit more anterior, and the morphine-sensitive neurons are a little bit more posterior. So we can see that there's a significant difference between the number of morphine, again, morphine ensembles, what this is saying, but that's just saying morphine-sensitive cells, and the saline-sensitive cells, depending on which time point we're talking about. So obviously right here, we can see that there's a lot more of these morphine-sensitive cells at, minus 3 millimeters from bregma. Okay, so the whole point of that is just to show that, indeed there is a population of cells that's sensitive specifically to morphine, and those neurons are the ones that we're going to be trying to characterize. Okay, so they're also going to do something called retrograde tract tracing. Okay, so what this is a toxin that they inject. Again, this all sounds very scary now that I'm thinking of people who are not in this world to be hearing all these words, viruses and toxins and shit. It sounds fucking terrible. But this is like the harmful aspects of all of these tools are essentially taken out before the researchers deal with them. So this is all like BCA, like BS level 1, like the lowest level of security bio fucking hazard shit you need to worry about when you're working with all this stuff. We do work with some rabies stuff later on, which I think is a little bit higher, but there are like various levels, like certain institutions and certain labs are not allowed to work with certain harmful stuff. So it's not like we get like another fucking COVID from CTB or from these AAVs, you know what I mean? Like this shit is really, really safe. So in any case, CTB stands for chlorotoxin subunit B, and the 488 is referring to the color of the protein that it's going to express. And the reason why they use this is because it has a property that makes it so it infects the axon terminals, which is the, part of the neuron that's projecting to some other brain region. You remember how I said there's principal neurons? And they originate in one area of the brain, like the VTA, but then they send collaterals to another area of the brain, in this case, the nucleus accumbens. So what they're going to do is that they're going to inject chlorotoxin subunit B with a green fluorescing protein into the nucleus accumbens. This is then going to be taken up by the axon terminals and travel retrogradely up the axon into the soma of the neuron. Okay, and that's important because they're also going to inject this CRHCeA into the VTA. So what that means is that you're going to have neurons that are co-labeled, meaning that they fluoresce green and they also fluoresce red when you look at them under a microscope. It's fucking amazing. Okay, like what I'm saying is, do you understand what I'm saying? These neurons are going to glow red and green. And when you see a red and a green neuron in this context, what it means is that it's a neuron that was sensitive to morphine and it is projecting to the nucleus accumbens, which, remember, is that fucking area of the brain that's super important for the reinforcing effects of drugs. Now, they're also going to use a technique called immunohistochemistry. Now, what this does is it allows you to do a third fucking staining. Okay, so this is all microscopy work. A third staining so you can see neurons that express tyrosine hydroxylase. Okay. So here are the representative images of these neurons. And, neuroscience, behavioral neuroscience is always pretty beautiful when you're looking, microscopy is always really fun to look at when you have all these cool fluorescing proteins and stuff. But essentially we can see what these look like. So here we have of those two fluorescing proteins. So we have the green one here, and when you see a green neuron, that means that these are neurons that are projecting to the nucleus accumbens because that's where this CTB is originating. Then when you see red neurons, that means that these are neurons that are sensitive to either saline or morphine, okay? Excuse me. And then these are probably far red, but they are represented as like gray or white. Excuse me. But these neurons are neurons that are positive for dopamine. Not dopamine specifically, but tyrosine hydroxylase. So we know that dopamine is there. And then here up at the top, you can just see that this is a representative image of the placement of the CTB. DAPI is just a fluorescing protein that selects for just basically cells. So it shows you the nucleus of a cell. And here we can see a track mark where they put the injector and then the spread of the virus. And then this little outline right here is the nucleus accumbens core in here and then shell out here. So it's in the nucleus accumbens. And so we know that when we see neurons that are in the VTA that are green, we know that those are neurons that were successfully expressing this CTB retrograde track tracer. Okay, cool. So now we get to some data that we can actually interpret here. And on the y-axis, we have NAC projecting ensemble percentage. And then on the x-axis, we have our groups. So it's really important to, whenever you're looking at these papers, that you identify. It's fucking geriatric. My back just decides to hurt for no reason at all. that identify the groups and what is it we're talking about. So this NAC projecting ensemble percentage, this means that we're not getting the raw numbers of the cells, but we're getting the percentage of the neurons. So the percentage of the neurons that we're projecting to the nucleus accumbens that were the morphine ensemble is higher than the saline ensemble, which means that when they inject morphine and they target those neurons that are sensitive to morphine, a higher percentage of them are projecting to the nucleus accumbens than the saline ensemble. Okay, so then when we look at the tyrosine hydroxylase positive NAC projecting ensemble, again, we see that there's significantly more of these in the morphine ensemble compared to the saline ensemble. So this is saying that essentially the morphine sensitive cells are dopamine cells that are projecting to the nucleus accumbens. So now what they want to do is something that's really fucking incredible, and it's using a tool called optogenetics. So unfortunately, this is a really complicated thing that I'm not going to go super in detail about, but what it is that it expresses a protein that's sensitive to light, a certain wavelength of light. And when you shine that light, so that's what this is trying to show here, when you shine the light on that protein, it causes that neuron that houses that protein to fire an action potential, which means activate the neuron, okay? So if your hypothesis is that these morphine-sensitive neurons are reinforcing, meaning that the animal likes the way it feels when those neurons are activated, then if you inject an optogenetic and activate that optogenetic protein, then what's going to happen is that the mouse, in this case it's a mouse, not a rat, the mouse is going to actually self-stimulate. Okay, so it's going to go over to the lever or like a little nose poke, and it's going to pull that lever, and that's going to activate the light that's shining, on this area of the brain and activate those neurons. So they're going to inject CRHCeA, which is again saying that in activated neurons, we're going to get Cre recombinase with an ERT2, this estrogen receptor on it. And then we're going to also put in this DIO, which means Cre dependent. And this is a channel rhodopsin, which is the optogenetic protein that I was talking about, and an M-cherry. So they're also going to have a control, which is just DIO M-cherry, which means that they have their optogenetic approach as well as just the fluorescing protein itself. And that is to control for the effect of Cre and, just causing recombination. Then 4 weeks later, they're going to inject tamoxifen. The next day, they're going to inject either saline or morphine. And that is going to cause either the expression of just M-cherry or the expression of this channel rhodopsin, which is, again, that optogenetic approach that I was talking about. And here we can, again, see representative images of what their viral expression looks like. We have here the VTA, and all of this red right here is the virus. So that means that some of this virus escaped the VTA, because we can see here that there are some positive cells outside of the VTA. I guess that's not too much of a problem. I don't know. And then here we have a sagittal section of the brain, and we can see kind of what the spread of this channel rhodopsin looks like. So I'm not super familiar with the VTA. But yeah, I don't know. This seems like kind of an amazing amount of spread for this kind of virus. And then also you have some down here. So I don't know. That seems like a little bit weird to me. Like, maybe, how do we know that this activity is localized to the VTA when it looks like there's so much of it everywhere? But in any case, they're then going to train the mouse in a Pavlovian conditioning procedure. So what that means is, or not Pavlovian, I'm sorry, it's actually Skinnerian, but a operant conditioning procedure where essentially the animal has to do a task in order to get a reinforcer. So it looks like they do 6 days of this, where the animal is pressing a lever, or probably, yeah, they're doing nose pokes since it's a mouse. They're going to be putting their nose into a little hole, and then that causes the laser to turn on, which is then going to activate these neurons that are either sensitive to saline or morphine. And then we'll be able to see if they like the way that feels, because if they do like the way that feels, then they're going to increase the activity. And then on days 7 and 8, they're going to give a vehicle injection. And then on day 8, they're going to give flupentixol, which is an antipsychotic, but the reason why they're using it is because it's a dopamine antagonist. So if they think that these neurons that are in the ventral tegmental area are reinforcing, then they should see activity. They should see the mouse pressing or doing the nose poke behavior through days 1 through 6. And then they have a further hypothesis, which is that these neurons are dopaminergic. So if that's the case, that when you give flupentixol, which is going to reduce dopamine activity, then we should see the nose poke behavior go down. So that's why that's happening. So here we can have this is their graphical abstract from the paper, where they basically are putting their nose into the little hole, and the light cue turns on, and then the laser is turned on, which then activates these neurons in their ventral tegmental area. And we can see very clearly here that these mice really like to stimulate that morphine ensemble, or the neurons that are sensitive to morphine. So let's look at our groups here. We have morphine ensemble channel rhodopsin, saline ensemble channel rhodopsin, and morphine ensemble M cherry. So remember, the M cherry is a control virus, just to make sure that anything that we've seen is not due to some fuckery with like the fluorescent protein causing an effect instead of the channel rhodopsin causing an effect. So that's a really important control that they included. They also have here the saline ensemble. So remember, when I say ensemble, I just mean neurons that are sensitive to saline, or neurons that are sensitive to morphine. So in here, if they stimulate the saline ensemble and they saw an increase in nose pokes, and that would be kind of bad for their hypothesis, because that would indicate that, it's not just morphine sensitive neurons, it's any neuron that is in the VTA that will cause this behavior. So that's really, really important, this finding. Sometimes people are like what kind of novel research is happening? Science is just fucking dying, and like nobody figures shit out anymore. It's like, it's actually really important to see neuron level specificity. Okay, because the canonical view in the past was that, well, look, if you just stimulate the ventral tegmental area, those are all dopamine neurons, and that's just going to cause activity or cause reinforcement. But no, when they stimulate neurons that are specifically sensitive to saline, which we did find that there is a population of these neurons in the very beginning of the paper, when they stimulate those with this channel rhodopsin approach, we can see the gray line here. Those mice aren't really nose poking. Okay, so they don't care. It's not significantly different from the group that just got no channel rhodopsin. Okay, so we have here several things to look at on this graph. So I'm probably jumping around too much. I'm making it way more confusing than it needs to be. So let's just start from the top. We have the y-axis here. And the y-axis is showing the number of times the mouse put its nose into a little hole to get the stimulation from the channel rhodopsin. On the x-axis, we have the number of training days. Okay, so this is day 1, day 2, day 3. Then we have our different groups on the active nose poke and our different groups on the inactive nose poke. So normally you have 2 nose pokes, and the mouse can poke one hole and get the reinforcer, and when the mouse pokes the other hole, nothing happens. And that's a really important control, because it might be the case that the mouse is just an idiot and likes to, poke, put his nose in the little holes and investigate. But we can see that that's not the case, because all of these triangles are all fled down in the bottom. The mice are not really poking the inactive nose holes. Importantly, the only group that is showing an increase in nose pokes is the group that had the morphine-sensitive cells targeted in the morphine ensemble and is getting that channel rhodopsin protein, okay? So this is indicating that specifically those morphine-sensitive neurons are reinforcing, because if they weren't, the mouse would not be putting its nose in the hole, it would be staying away from the hole because it either doesn't learn or it doesn't like the way it feels. And we'll see that later on in the paper, I do believe. So here they're also showing that these neurons are dopaminergic because when they give a dopamine antagonist, they significantly reduce the number of nose pokes that are occurring on day 7 to day 8. So here we can clearly see that the mice like the way it feels, and when you give them something that blocks dopamine activity, their nose pokes go way down. So now they're going to implement another really cool but complicated tool called fiber photometry. So what this allows the researchers to do is to look at calcium activity in the neurons. And what that tells you is when a neuron is activated. So they're going to put this into the nucleus accumbens, and they're going to see, are these neurons activated when we stimulate neurons in the VTA that are morphine sensitive cells, okay? So that's what this is kind of showing here, is that they're going to stimulate cells in the VTA, but then they're going to see if neurons in the nucleus accumbens are activated. And if they are, then you can make the inference that the reason why they're activated is because of this direct activity that you're causing in the VTA. Okay, so here are some representative traces of the two groups. We have our saline sensitive neurons here and our morphine sensitive neurons here. And then the green is the fluorescing protein that's associated with the fiber photometry. For anyone that's interested, it's an HSyn DA4. So what this basically means is that like in neurons, we're going to be looking at dopamine activity. So I said calcium earlier, but it's because normally it is looking at calcium influx or efflux, but essentially this is going to be looking at dopamine activity. So I hate heat maps, but heat maps have gotten very popular in neuroscience. I'm a bigger fan of just line graphs and bar graphs, so I'm just going to get those on the screen. But basically, this heat map is showing the activity of the neurons, and we can see that the redder or the hotter colors are more active. So we can see that like in the morphine ensemble, it's just, you can see clearly from the heat map that there's a lot more activity going on here. We can also see that the change in fluorescence, so this is a delta F over F, so that means that the change in fluorescence over fluorescence is higher in the morphine ensemble as soon as the laser gets turned on. So that's really important. So when they stimulate these VTA neurons, that's when we get the change in fluorescence. That's when we get the release of dopamine. And then we can also see that when they're quantifying this data as the area under the curve. Again, like what all this means is like not super important. What you can take home is that the morphine sensitive neurons are much more activated in releasing dopamine when you stimulate them compared to the saline sensitive neurons. And again, it seems like you might say like, well, obviously that's the case. Like, why would we expect anything different? Because the canonical view is that when you stimulate the brain region, that's where you're going to get all the activity, and it shouldn't be neuronally specific. But these really advanced tools are showing us that it's not just the VTA. Okay, it's not just this whole group of neurons. It's specifically these neurons that are sensitive to the pharmacological activity of morphine. It's very cool. So, oh my God, this is only figure 1? How many fucking hours is this going to be? Yeah, this is, I think when I did this originally, it was like a 45-minute presentation. So I have no idea how long I've been recording, but it's way too fucking long. So figure 1 conclusion. Morphine cells are in the medial VTA. They project to the nucleus accumbens and are dopaminergic, and activation of these cells is rewarding. Okay, so this is fucking very good, cool thing. So the next figure is going to try and show us that chronic morphine enhances inhibitory synaptic transmission onto the morphine ensemble, and this is involved in the development of the negative effect during opiate withdrawal. Okay. So I wrote that like 3 years ago, so I just read it to us, but I'm sure that that's what we're going to find right now. Okay, so again, they're going to use the same approach here. They're going to have CRHCeA, so this is going to allow them to select for neurons that are sensitive to saline or morphine, and those neurons are going to express a red fluorescent protein so that we can see them in the microscope. Now, What's going to be different here is that this is an ePhys or an electrophysiology experiment. So this is really complicated. The electrophysiologist, so I'm a behavioral neuroscientist, which means that like primarily what I'm focused on is the behavior. I do a lot of microscopy and I do a lot of ways like of affecting the behavior. So I put like in my own research, I put the viruses into specific brain regions and then, either activate neurons or inactivate neurons. But what I'm primarily focused on is the actual behavioral output of the, experiment. Whereas an electrophysiologist is more interested in the actual behavior of the cells themselves. Okay, so it's really complicated, and I'm not going to be able to fully explain. It's not going to be super intuitive what is done here, but essentially what electrophysiologists do is that they have a glass pipette, which a pipette is like this little, it's a little cylinder glass, thing, for lack of a better term, that has artificial cerebrospinal fluid in it. And essentially, they're going to patch onto, I'm sorry, they're not putting that in the glass pipette. The glass pipette is being lowered into a solution that's full of it. But anyways, this glass pipette has a tip that is so small that it can actually puncture the surface of the neuron and make a contiguous seal with the neuron and the pipette. And then they can actually measure the electrical activity that's happening in the neuron. Okay, so it's kind of complicated, but just know that like what electrophysiology is doing is that it's saying, okay, what's happening to the neuron? Is it firing or is it not firing? That's normally, or is it getting excitatory signals or is it getting negative inhibitory signals? That's normally what electrophysiologists are doing. So what they're going to do is see if repeated morphine injections are changing the physiological properties of the neurons themselves. Okay, so they're going to do an electrophysiology recording on day 1, and then they're going to give repeated doses of morphine to the mouse in an increasing dose over days. Okay, and then on day 7, they're going to do another recording, and they're going to see if the electrical behavior of the cells has changed. It's really fucking amazing. So here we have some representative images of what the fluorophores look like. So we have an M-cherry neuron, a biocytin neuron, and tyrosine hydroxylase. So essentially, this is how they're going to find those neurons that are dopaminergic and are sensitive to the morphine, basically. So now, I will tell you that most people, even at the graduate level, are going to have trouble with these kinds of graphs because electrophysiology is really complicated. So you may have to take my word for it a little bit on this, but basically these things right here are representative traces of the recordings of the electrical activity on the neuron. Okay, so this is data, but it's not really what we care about. This is just a representative trace, so we can see what it looked like when they were recording from the cells. Here is the data quantified. Now, we have in gray the neurons that were sensitive to saline, and in maroon or whatever this color is, the neurons that were sensitive to morphine. And this is a cumulative probability plot. So essentially, this is taking into account all the events that were measured and then looking at what percentage of those events, if the percentage of probability of those events occurring is different in one of the groups. So we can see that when we're looking at what's called miniature inhibitory postsynaptic currents, okay, it's such a fucking nightmare, but this is looking at the inhibitory signals that is coming into the neuron. So remember how I talked about GABAergic interneurons and how they cause inhibition? That's essentially what this is looking at. It's not looking at GABA, but it's looking at the inhibitory signals that the neuron is getting. And to make this even more confusing, when you're looking at these kind of currents, this is called an outward current, which means inhibitory. So that's why they're showing the representative trace, is to, you know, verify to people that do this kind of stuff, which is not me, by the way, that like, yeah, indeed we're looking at, you know, outward currents, which are inhibitory, or inward currents, which is this one, which means excitatory. So it seems a little bit backwards and non-intuitive if you're not an EPHIS person. But basically, when we see these big spikes, that's saying, okay, the neuron is getting some kind of inhibitory signal right now. So we have a leftward shift in the saline, or in the morphine ensemble, with the amount of the inhibitory signals that they're getting. So the speed at which the neurons that are sensitive to morphine are getting inhibitory signals is increasing. So that's what this inter-event interval is saying. It's the time in between the events. That's what the inter-event interval always is referring to. But in this case, the event in question is a miniature inhibitory postsynaptic current. So the speed that these morphine ensembles are being inhibited is increasing. Okay? And also the amplitude of the inhibitory current is also increasing. So we can see that the inter-event interval is shifting to the left, which means that it's happening more frequently. And we can see that here quantified as frequency. So the frequency of the inhibitory events is increasing, and the amplitude, which is just, I'm going to say in like a, you know, as a person that's not a biophysicist, I'm just going to dub it down for the rest of us, which is to say that, you know, the strength of this inhibitory signal is increasing. So they're measuring this in picoamps. The probability of there being an increased amplitude is increasing in the morphine-sensitive neurons. Yeah, and so we can see the amplitude here, you know, in this bar graph is a little bit more intuitive. It would be, I mean, they don't have the bars, but basically this is what like a bar graph would look like. It's a scatter plot. But regardless, yeah, so we can see that the inhibitory postsynaptic currents are changing. Now that's important because when we get back to our little diagram of what the paper is trying to explain, or if you go back in the video right now and you look at that, you'll see that part of their hypothesis was that there's an increase in inhibition towards the morphine ensemble. And then we can also see here that the miniature excitatory postsynaptic currents are not changed between the two groups, despite the treatment of morphine. Okay, so that is actually super important to the overarching hypothesis of this paper. Now, here are some terribly stupid diagrams that I made because I was afraid that people in my graduate seminar weren't going to know what these behavioral procedures were. But this one over here is condition place preference. In this case, they're going to do a CPA conditioning, which is condition place aversion. But regardless, it's a similar concept. When you see CPP or CPA, it's that it is a chamber, and you can't see it right here, but there's a wall separating the two chambers. And they will put the rodent in one of the chambers and give it a treatment repeatedly. In this case, they're going to be stimulating either the morphine ensemble or the saline ensemble with an inhibitory dread. Oh my God, I haven't even explained dreads yet. This is why this kind of thing, like when you're talking to people that are not familiar with the subject, it's like it's going to take, and this is not me trying to be condescending or anything, it's just that like when people say go do your own research, it's like the reason why that's kind of a stupid thing to say when you're talking about really high level scientific endeavors is because, I mean, you have to get a whole undergraduate level. I mean, most undergraduates who have a degree in behavioral neuroscience are not going to be familiar with a lot of this stuff, to be honest with you. I've just talked to a lot of undergrads, and, you know, a lot of good students will be familiar with this, but if I said, do you know what an inhibitory dread is or an HM4DI to, you know, a sophomore, well, definitely not a sophomore, but like a senior that is studying behavioral neuroscience supposedly in college, like they won't even know. So, in any case, it's just so complicated. But dreads have a very cool name but it's an acronym that stands for designer receptors exclusively activated by designer drugs. They are a synthetic or I don't want to say fake because they're real, but they're a synthetic receptor that is modeled after the human muscarinic receptor. So that's what the HM is standing for. And they are a G protein coupled receptor, and they're coupled to an inhibitory protein. So when you have this, what this means is that we have a cre-dependent inhibitory dread that expresses mCherry. So in neurons that are going to be sensitive to morphine or sensitive to saline, we're going to get this inhibitory dread. And that means that you can inject another drug that will activate this receptor. And in this case, activating this receptor means that you're going to get a decrease in neuronal activity. So what they're going to do is that they hypothesize that the neurons that are sensitive to morphine are sensitive to or are reinforcing. So if you inhibit those neurons, right, then you should get aversive behaviors. That's the hypothesis. So you can see that it's like in figure 1, they show that the mice, so I'm showing rats here because I work with rats, but these people are working with mice. So disregard the fact that these are rats and not mice. But in any case, they showed in figure 1, if you remember, that stimulating these neurons is reinforcing because the rodents want to keep doing it. But if you inhibit the neurons, which is, again, a part of the hypothesis of this paper, is that repeated activity of these cells results in a decreased overall activity during withdrawal. So if that's the case, then presumably you should be able to inhibit the neuron. And if your hypothesis is correct, then we would predict that there would be a decrease or that there would be an aversion that would be associated with inhibiting these cells. And that's what they're going to test with this first box here, this conditioned place aversion test. Now, this other one is called an elevated plus maze. Now, this is a measure of anxiety because rodents don't like to be near high places. So if a rodent is less anxious, then they are exploratory though. They really like to explore. So if a rodent is less anxious, then they will go hang out on the open side of the elevated plus maze, and they will explore, and they won't, they won't be tripping about the fact that they're kind of up in the air. If a rodent is more anxious, then they spend more time in the closed arms. So this is just typical rodent behavior. They want to get to the sides. They want to get away from the light. They want to get away from the height. So in this experiment, they are going to condition these rodents to the CPA conditioning. So they're going to put the rats in one chamber of this, and then they're going to repeatedly inhibit those morphine-sensitive neurons. And then they're going to see which side of the box the rat spends more time on. And that's indicative of either a conditioned place preference or a conditioned place aversion. Then they're going to, at the end of this, they're going to do an OFT, which is an open field test. I don't think that they show it because there's no effect. So they keep that in the supplements, the supplemental figures, which is like the second part of the paper. Or the hidden part of the paper. It's not hidden, actually. You can go find it. But it has a lot of controls oftentimes. And they're going to do the elevated plus maze. And CNO is the drug that's going to be activating this inhibitory dread. Okay, so this graph right here, the CPA score, is talking about the behavior in this box. And we can see that when the rats are initially put into the box, they spend an equal amount of time on both sides of the box. But once they get that morphine ensemble inhibited repeatedly, then they spend less time on the side of the box that was associated with inhibiting this morphine ensemble. So it's kind of like if you, every day were going to a particular environment and like you kept on getting like hit in the face, you would avoid that environment, right? Because you're like, fuck, like this is like not a cool place to be. So that's essentially what this is modeling, is that like if the rat doesn't like the way that it feels to be on one side of the box, then they will avoid it because they'll learn that that side of the box is aversive. And similarly, here we have the elevated plus maze, and we can see a significant decrease in the time spent in the open arms. So what that is indicating is an increase in anxiety in the rats that got the morphine ensemble inhibited. Now, there's no perfect behavioral assay, and it is the case that like sometimes these behavioral assays are like really criticized because are we anthropomorphizing the rats and the mice? How do we know what they're thinking and feeling? And those are all valid, but it's about the preponderance of evidence that makes us come to the conclusion. I mean, you wouldn't just expect that just because there's an effect in the elevated plus maze that you know for sure what is happening here. It's because of all the other information that we've so far gotten, that when you activate these neurons, the mice like it, and the fact that we've seen what's happened on a physiological level to the cells, and the fact that when you inhibit these neurons, the mice are avoiding that chamber. It's all of these things that make us think, okay, I think we can come to some conclusion about this. Now, they're going to repeat this experiment, except for now they're going to put the mice into a withdrawal state, okay? Because again, remember their hypothesis really relies on repeated exposure of these neurons in withdrawal will result in aversive feelings, okay? So they're going to label the neurons, again, either the saline sensitive ones or the morphine sensitive ones, and this time they're going to use an excitatory dread. So in this last experiment, they used an inhibitory dread. That's an HM4DI. In this experiment, they're using an HM3DQ, and that's an excitatory dread. So that means that when they activate this protein, it's going to cause the neuron to fire a lot. So it's similar to in the first experiment when we showed you that excitatory optogenetic approach. This is very similar to that. Instead of using a light, though, it's using a pharmacological agent, in this case, CNO, which is not important what it stands for. Okay, so they first label these neurons, and then they are going to put them, they're going to be giving them a pretest in the condition place preference box, and then they're going to be giving them increasing doses of morphine. And then they're going to put the mice into a rapid withdrawal state, and that's going to cause a condition place aversion. Sorry, it's been a while since I've read this, so I was just trying to figure out what is happening here. But essentially, they're going to give 3 injections of CNO during a CPA conditioning. So what that means is that the mice are going to likely be going into withdrawal here. So essentially, there's a pretty characteristic time point when you give the injection of the drug and then when you expect to see the mice going into withdrawal from not having the drug. So it looks like on day 6, they're going to do 3 consecutive sessions of the mice who have been getting increased doses of morphine into the box, and then they're going to be inducing that withdrawal, or the mice are going to be going into withdrawal, and then that's going to cause an aversion, and then they're going to see if activating these neurons with this HM3DQ is going to minimize some of that withdrawal aversion. So they're actually both using the HM4DI and the HM3DQ. So they're basically going to replicate this first experiment and add to it with this excitatory dread. So in the first experiment here, what we can see... Oh, okay. Sorry. Yeah. So it's because they're going into the withdrawal in this one and they didn't in the first one. Sorry, it's been a while since I've read this. So yeah, so what's happening here is they give them the repeated injections of morphine, they put them into withdrawal, and then they also give them CNO. And what we can see here is that the mice are both developing a conditioned aversion to the context that's associated with the withdrawal. But that aversion is increased in the mice that are getting those neurons inhibited even further. Okay, so the inference here is that since both of them are experiencing withdrawal, there's inhibition to those cells, but if you further drive inhibition with this inhibitory dread in the morphine ensemble, then that's going to cause even more aversion. And that's kind of what they're trying to show here, is that although they both both groups are showing a significant aversion to the context. The mice that are getting that morphine, those morphine sensitive neurons inhibited, are even more averse to the context. And similarly, we can see that there's a decrease in time spent in the open arm of an elevated plus maze, so even more anxious. Now, here is a very interesting effect. We can see here that the mice, if you activate that morphine-sensitive group of neurons in the VTA, that they are slightly less averse to the context that was associated with the withdrawal. Okay, so whenever you're like kind of listening to these kind of presentations, it's really difficult to like pay attention sometimes, because like, especially if you're not familiar with it, you'll just start zoning out, and then if you miss like 30 seconds of what the person was talking about, like you're totally lost. But it's always important to look for the pattern. And here we can see a very interesting pattern in O and R. So O is this graph and R is this graph. And we can see that the gray saline ensemble bar here is inverse from this one, right? So it's like we have the red one, the red bar is going lower than this gray bar here, but it's higher than this gray bar here. Okay, so that is, should be a clue to you that essentially what we're getting in O and R are inverse effects, okay? Which means that when you excite these neurons, it's protective against the withdrawal symptoms. But when you further inhibit them, it makes you even more vulnerable to the negative aspects of the morphine withdrawal. And then again, also we can see an increase in time spent in the open arm, which is indicative of a decrease in anxiety. So so this is all going really nicely together with trying to build this story that when you're in morphine withdrawal, there's an inhibition of dopamine neurons, which is then resulting in the negative affective state that is associated with this withdrawal. So what they found out in figure 2 is that chronic morphine inhibits those morphine sensitive cells, okay? So over time, when you're taking opiates over and over again, those neurons are becoming more inhibited. And that makes sense from a homeostatic point of view, is because those neurons are going to be, exogenously activated repeatedly. And so your body is going to, or your brain is going to try and be like, hey, what the fuck? You guys aren't supposed to be activated this much. So there's some mechanism that's kicking in to say, you guys relax. So, they also show that inhibition of these cells is aversive. Okay, so I didn't show the rest of the graphs, but the take home of this is that while activating those neurons is very reinforcing, as we saw in the first figure with the mice self-stimulating those neurons, if you inhibit them, they're actually really aversive. And also the problem with that is that chronic morphine causes inhibition of the cells. So you can see where this could potentially cause a cycle of not good stuff to be happening. Okay, so we've gotten half of the thesis kind of shown to us here. So the next thing that they're going to be doing here is trying to tie in the central nucleus of the amygdala and those CRH neurons. So this next figure is going to suggest that the activation of corticotropin-releasing hormone central nucleus of the amygdala neurons that project to VTA neurons facilitates CPA and anxiety during opiate withdrawal. Oh, man. Yeah, so this is complicated but fun. So essentially, what they're doing, again, I haven't seen these figures in like 2-3 years, whatever. But okay, so what they did here was they injected a retro-cre-dependent yellow fluorescing protein, and it shows up as green on the pictures, but it's called yellow. So it's because it has to do with the wavelength, but don't worry about it. So in any case, they inject this virus into the VTA, and it's cre-dependent. And they're using a transgenic Cree mouse, okay? So this means that in this mouse, it was bred and it has genetic manipulations to it. So that's why it's called transgenic. That is going to express Cre recombinase in neurons that also express corticotropin releasing hormone. Okay, so this is super important because this is how we're going to tie in the CRH part of this. So they're going to inject morphine over the course of days, and they've already shown that this injection protocol results in an increase in inhibition towards these VTA neurons. So we already know that this morphine IP injection protocol is a good way to put these mice into withdrawal, basically, and to get them addicted to opiate. And also, we can see here that they're going to wait 10 hours and then do what's called CFOS staining. And so this means that it must take about 10 hours for the mice to go into withdrawal. So CFOS is an immediate early gene that expresses a protein after neurons have been activated. So basically, when a neuron fires an action potential, there's some period of time, it's about 90 minutes, and then you'll get the expression of the protein called FOS. So FOS is doing a lot of things other than what we care about, but the reason why they're measuring FOS is because it is showing which neurons are being activated at a particular time point. Okay, so what they're going to do is that they want to look at the overlap of neurons that are expressing FOS and expressing CRH, because that will give us some clue of which CRH neurons are activated as a result of morphine withdrawal. Okay, so here on graph C, we can see on the y-axis CFOS co-localized percentage. So what that is saying is that in these representative images, they're looking at the number of neurons that expressed FOS and were expressing CRH. And we can see that by far, it looks like the neurons in the central nucleus of the amygdala have the highest level of activity that are also expressing CRH. They also looked at the BNST, which is the bed nucleus of the stria terminalis, and the PVN, which is the paraventricular nucleus of the hypothalamus, I believe. But basically, we can see that in the BNST, there is some significant difference here between the morphine withdrawal and the saline animals. But it does look like it's a bit smaller than what's happening in the central nucleus of the amygdala. And in the CEA, we can see that there's an increase, a significant difference between the saline and the morphine withdrawal animals, which is important. So that's why they're going to be focusing primarily on the CEA, or the central nucleus of the amygdala. They probably... Again, I haven't read the paper in a long time, so in the paper they would explain what I'm about to say, but basically they probably have some other data where they looked at the BNST, the bed nucleus of the stria terminalis, and they probably didn't find anything, so that's why they're going to focus primarily on the central nucleus of the amygdala from here on out. And in the PVN, they just didn't even see anything. Now, why did they choose to look at these regions in particular? I don't know very much about CRH, but if I hazard to guess, what I would say is that these are likely areas that are known to express CRH. So I don't know where CRH is expressed, if it's expressed in other areas other than these three, or what, but basically that's probably why they chose these three. So they're going to, again, now focus only on CEA neurons, and specifically neurons in the amygdala that are CRH positive, okay? So again, they're going to be using this, in order to do that, they're going to be using a Cre-dependent retro YFP. So that means that they're going to inject the retro YFP into the VTA, and that's going to make neurons that are CRH positive in the amygdala fluoresce green. Then what they're going to do is that they're going to, again, look at the electrical activity of these neurons when the mice are in withdrawal. So 4 weeks before the experiment, they do the injection of the virus, and then they start giving them. They do one cohort that's getting looked at on day 1, and then another cohort that's getting looked at day 7. And in between this, they're giving increasing doses of morphine. So one thing that's a little bit of a weakness to this is that, like, in order to do EPHIS, you have to euthanize the animal, obviously. So although they are looking at day 1 and then comparing it to day 7, you can't compare within subjects because all the mice that are killed on day 1 obviously cannot be preserved to day 7 after getting this treatment protocol, right? So that's one weakness to this. It would be stronger to have a within design if you could look at the neurons on day 1 and look at the neurons on day 7. But given the what is happening with EPHIS, the fact that you have to isolate the cells, it requires the brain. So requires the brain to come out, right? So here what we can see is that, again, we have these cumulative probability plots that we looked at previously, and they're looking at the inter-event interval and the amplitude of miniature excitatory postsynaptic currents. So again, this is looking at the amount of activity that's occurring in these levels, like the excitatory activity. So we can see that the speed or the frequency at which these neurons are being activated on day 7 is significantly higher than how they're being activated on day 1. And then we can also see in the next graph that the strength of this activity is increasing on day 7 relative to day 1. So what the inference here is that morphine is doing something that increases excitation onto these amygdala neurons. Again, here's some representative images. I noticed I didn't look at it, but this is actually, it's kind of hard to see this, but basically these are neurons, and this is what it looks like under the microscope when the researchers are doing EPHIS. So here they're showing DIC is the kind of microscope that's typically used when the researcher is patching onto neurons. And it's really kind of complicated how this works. I think that this is honestly a really bad picture to try to show somebody like what EPHIS looks like. So don't judge it too harshly based on this because it's really hard to see. I think this is the pipette basically coming down right here, but I can't be certain because the picture is too small and kind of low quality. But in any case, we can also see here that the spike number, so that's the number of action potentials that's actually occurring, is increasing dependent upon the current that's being injected as measured by picoamps. So essentially, they can force a neuron to have an action potential. And the number of action potentials that happens as a result of this particular amount of current is higher in the day 7 neurons compared to the day 1 neurons. So that means that it's like the neurons are more excitable, basically, is what all this is trying to tell us. Okay, so what do we have here? Something fucking complicated, let me tell you that much. So we have a Dio Inferno is what I always call this, but YFP. So this is another optogenetic approach. And they're essentially going to be injecting this excitatory opsin into the nucleus accumbens. Into the amygdala in the CRH mouse. So what this means is that in the amygdala and in neurons that are CRH positive, there's going to be the expression of that optogenetic protein that we talked about previously. Now, that protein is going to be expressed in the soma, but it's also going to be expressed in the terminals of these neurons. So what that means is that you can shine the light just at the VTA and isolate the neurons that you care about isolating. So what that means is that you can specifically target these CRH neurons that are only projecting to the VTA. So that's a super important ability because the amygdala is probably, it definitely is projecting to a lot of different areas that they're not showing right here. But you know that you're only affecting the VTA neurons because you're shining the light only on the VTA spot. Okay, so here's their experimental, and this is just looking at the, this is a control. So they're showing that they did some ephys on some of the stuff to validate the optogenetics. So they can show that when they have the laser going, it is activating a Inferno. Oh, okay. So this is inhibitory. My bad. So this is a halorhodopsin. So it's inhibitory. So they can show that basically when they are firing, when they're activating this inhibitory opsin, that it's causing suppression of activity in these neurons. So then they're also going to put these mice into withdrawal and then inhibit the CEA neurons. Oh my god, this is so fucking cool. So do you guys do you understand the inference? So basically, again, I haven't read this in a long time, so I'm kind of like rediscovering it with you. But basically, what is probably going to happen here is that they're going to show that if it is the case that these neurons are aversive, then inhibiting them should reduce the aversive nature of the morphine withdrawal. Okay, so that's the whole point of this experiment, is that first they showed, look at how reinforcing these neurons are. And then they showed with DREADS that, if you are targeting these VTA neurons, that you can inhibit the withdrawal. But now they're making a greater claim, because they're saying not only is it the neurons that are in the VTA, but it's specifically these CRH neurons that originate in the central nucleus of the amygdala that's causing that effect that we saw previously. And indeed, that's exactly what we see. Okay, beautiful. So we can see that the CPA score here, again, remember, they're going to be spending less time in the chamber that's associated with the negative feelings, right? So we can see that when they inhibit those neurons, they just totally abolish the CPA. In the YFP group, this is the control, meaning that it's just a yellow fluorescing protein. And we can see that in the control group, they develop a clear CPA. In the group that's getting the neurons inhibited with that inhibitory opsin, we can see that it's abolished. Okay, so that's great. Also, we can clearly see here in the group that's getting those amygdala neurons silenced, the mice are spending a lot more time in the open arms than in their control counterparts. So you can just see what this blue stuff is how the computer records how much time the mouse is spending in a particular area of, the chamber. And so here you can see that the mice do not like to investigate those open arms. They're like, fuck that noise, I don't want to fall off or whatever. But when you inhibit those aversive neurons, the mice are much more brave or less anxious, whatever you want to say. And they go out and they investigate a lot more. So that's super cool. I'm going to have to take a break and finish this later, unfortunately. How much more figure 3 do we have? Only one more. Okay, so I'll finish figure 3 and then I'm going to have to take a break because I have a appointment that I got to get to. Okay, so now what they're going to do is they're going to show exactly the inverse of this. So now they're going to put the Cre-dependent excitatory opsin in the VTA. In the central nucleus of the amygdala, and then they're going to shine a blue light onto the VTA to excite those amygdala neurons. Excuse me. So, let's see, what are we doing here? So this is just looking at, this is their control showing that they can actually activate neurons with blue light pulsing. So that's what this is showing here. And here's their experimental paradigm. So on day 1, they have the pretest for the CPA. On day 2, 3, and 4, they're doing conditioning with the laser. So they're going to actually activate these amygdala neurons that are in the VTA, or projecting to the VTA rather, on these 3 days. And then they're going to do the test, and they're going to do an open field test and elevated plus maze. And here we can see the exact opposite. Remember when I said to look for patterns? You can see that there's the exact opposite of O is what we see in U, right? So the mice that are in the control group, if anything, it looks like they're kind of having a slight preference, which is a little bit of a problem, to be honest. I don't know if that's significantly different from their pretest? Probably isn't. So it's not like a real problem, but you don't want to see that. You want to see shit that looks more like this. But regardless, though, because like realistically, it's just these two mice that are making it not a preference. If these two data points were not here, then this would be a preference. So that's not ideal. But who knows why that happened? Sometimes behavior can be a little bit wonky like that. But importantly, though, there's no denying this, which is that when they excite these CEA neurons that are projecting to the ventral tegmental area, that there's clearly a CPA that's produced that's different from this control group. And additionally, you can see the exact opposite pattern in the elevated plus maze that we saw previously. So in this one, when they inhibit those neurons, we can see that blue line, they're starting to get more brave and investigate. And in this one, we can see that when they activate those neurons, they're like, fuck that, we're not going out into the open arms anymore. Okay, so figure 3 conclusion is that morphine withdrawal activates neurons in the central nucleus of the amygdala, and that this increases excitability of these neurons. And additionally, these neurons are necessary for the aversive response, it seems to be, and sufficient to drive the aversive response. So necessary and sufficient conditions are extremely important in mechanistic studies, trying to show that not only if you inhibit the neurons does the behavior go away, that shows necessity, but also you can show that they're sufficient by activating those neurons and driving the behavior that you're interested in. So wow. So far, this paper has been super insane. How many figures are there total in this? Figure 4 is kind of long. Figure 5. And then there's a 6th figure. Whoa, and that one is complicated. Okay, cool. All right, so there's that. So there's 6 figures. We just got through 3 figures. I have an appointment that I got to get to. I didn't think it would be taking me this long, but this is a fucking super complicated paper. Lots of really fun stuff in this. So, I don't know if I'm going to put this on the same video or just make a part 2, I guess. So I'll see you guys in the next one.